And our next speaker, Daniel Garcia Parraga. He's a veterinarian since 2001, and he trained in several European and American zoos and aquaria. And then he ended up in the oceanographic in 2002 as a chief vet. Daniel is also involved in research, not only captive research, but also in field, like in Antarctica with seals, and with the wild animals, with the recuperation center that they have here. And he told me yesterday that he will present a paper and he feels like MacGyver. So I'm very looking forward to this paper.
Okay, thank you very much, Claudia. Good morning, everyone. I'm going to talk just a little bit about the importance of creativity and proactivity working as a veterinarian in an aquarium environment. As an introduction, we probably will know that knowledge is very scarce and fragmented related to medicine in this field. There are very few bibliographic references. Hopefully they are growing very fast, but still up to now the knowledge is very limited. And most of the knowledge we have about fish medicine and invertebrates medicine is based on aquaculture species for human consumption. So, on the other hand, we have a very extreme variability of species in our collection and very, very limited approved drugs or medications, diagnostic protocols for our species. So we have to figure out how to use other fields of the veterinary world in our species, because we have very, very limited treatments for our specific patients. This brings us to use the same treatments for a very diverse group, not only diverse in the species we manage, but also in the diseases or the problems we come across. Here you have just a basic diagram showing how is the knowledge about medicine, the general knowledge, how is it distributed. You see that human knowledge is still the big part, while veterinary knowledge is much more limited. But veterinary knowledge still is very, very focused or based on production, animal production, and companion animals, so domestic animals. And we see in the veterinary cake knowledge, the part of the aquatics, ornamental aquatics, I'll say, it's very, very limited what we know. And this limitation is not only about the knowledge, but also about what is regarding this world. When you want to become a veterinarian of aquatic or fish, or ornamental fish or ornamental invertebrates or marine mammals, we really have to struggle because we don't have precise programs at the university, so it's very difficult to find information and the formation to get into this world. So many times we have to work through trial and error. We do have, as I mentioned, a lot of difficulties for formal education, and sometimes it's really frustrating and challenging. So I have always in my office the stress reduction kit to go to it when the problem is going too complicated. But on the other hand, it's a very, very motivating world. It allows for your self-development, to do research, and to go fast, advancing fast, because we are really, really at the beginning. So our objective as a veterinarian in an aquarium is try to resolve the problem, of course, with the knowledge available, the resources available, and of course the hard nucleus of this talk, the ingenuity and creativity that definitely we need for this purpose. And of course, share experiences and results with other colleagues, so we'll have the feedback from them as well. As we mentioned before, Eva or Lisbon Aquarium or in SeaWorld or Claudia, we always are in contact, but we share our experiences. But sometimes it's true that none of us has time to publish, and many times this knowledge stays in very small groups of people. We do need to find time for publish, to attend these kind of conferences, and to share the knowledge a little bit farther than just the small team of people or the small friends group. Talking about materials, here at the aquarium, we do have the advantage of have a nice laboratory and veterinary infrastructure, mainly because we have a big collection of marine mammals, as you probably know. But we take advantage of all this equipment to implement techniques in fish and invertebrates, of course. We do have a human team, very motivated, and very important part as well is the trust that we should receive from the direction. I mean, it's very difficult many times to do things because the knowledge is limited, but you need to advance, and you really need the trust of the direction, curators, or direction of the park, Pablo in this case, I guess, to be able to keep on moving and, okay, let's assume certain risks to keep on moving. Talking about methods, what we use to do our job. Of course, bibliography, limited, but it's available. Internet, very useful, but very dangerous. So be sure that the information you are getting from it is trustful. Personal communications, of course, as we mentioned, we have many colleagues, and it's very important to communicate with colleagues. And of course, trials and error. Sometimes you are going to be the first one coming across the situation, so you have to find your way. But the most important part, and what I want to talk in this part, is the implementation of present techniques. We do have techniques, advanced techniques, very well designed for other species, mainly dogs, cats, and horses, and medications. But we do need to find a way of applying it in our collection. And on the other hand, we do have other techniques, not for medical purposes, but we can adapt for medical purposes, like, as we saw, the glue for dental prostheses, or the spear gun for fishing, to use it to inject instead of fishing the animals. So creativity here is taking the big part. Medicine, it's really an art. And here, just to say that limitations live only in our minds, but if we use our imagination, our possibilities really become limitless. Just here I mentioned some personal requirements to be creative, but this is not only applicable to the veterinarian in an aquarium, but I think that someone that is working in this fast developing environment should be creative. I'm going to share with you some results about how we try to solve in an unusual way the main limitations we find at the aquarium. For instance, how to retain locally a topical treatment or bandage. It's in the literature, it's already said that ore-base stays in the water, but it talks mainly, in the bibliography is referred as a paste. 6 years ago, we started to use the dental glue to cover the treatments that we were putting on wounds. And nowadays, we give a step forward, and what we are doing is mixing this glue, not only covering the treatment that we put in the wound, but also mixing this glue in powder with antibiotics and healing promoters like vitamin A or plant extract like aloe vera or Asiatic centei to really improve healing. Honey, I mean, it's a natural remedy that has been proved to work very well in humans and in other terrestrial species. And in our experience, especially for very refractive wounds that do not heal with anything, or even for thin rot, honey works pretty well without requiring antibiotics or other stuff. And in just a little bit more advanced, the hydrocolloid patches that are used for torpid ulcers in humans, for instance, especially in benthonic fish, elasmorangs mainly, that do tend to create too productive scarring tissue. We do cover this kind of lesions with patches when we have to do the surgery and they are in the ventral part of the animal to avoid this kind of proliferative cicatrization. We cover the lesions with patches with hydrocolloid gel, and it heals up very well. How to catch alive, of course alive, to treat a fish just individually in a big tank. We presented the adaptation of this pneumatic spear gun in another conference, and right now we implement even a little bit more the technique, allowing us to use different size of darts to inject larger volumes and connect it with this floater to the needle of the dart to be able to retrieve the dart, because if we have to anesthetize the fish, we are going to receive the fish and we are going to take the dart by handle. But in case we have to inject only antibiotics into a shark, if you dart him, many times the animal will retain the dart for 4 or 5 days. We don't want that. So we connect the floater to the dart and we are able to retrieve the dart without trouble. Here you have a case, for instance, of a shark that developed this kind of ulcers in the tail sac. And that's the typical case where we use our DAT gun because the animal was not eating, so we cannot medicate in another way. We'll have to cut the shark otherwise. Of course, parasites, as Anna was mentioning. Many times we do have problems with parasites, and the problem is bigger as bigger is the aquarium. So in very large tank, you all know that many people has to spend a lot of money to treat that. We do need to find treatments that are not polluting so much the tank and are efficient enough and much more economic. We try to use, for instance, at the beginning of the aquarium, to use caprylic acid to control cryptocariosis with, not fantastic result, I would say. We started at the beginning as well in 2003 to use luferunon. That's an information I found in the internet, trying to cope with Argulus problems. And I found in internet that luferunon was very useful, and I found in internet that the luferunon was very toxic. So that's what I mentioned, that internet information you have to. We did some trials in the quarantine with it, and it works very well. And nowadays we are trying to use other drugs like nitempiran. Luferunon and nitempiran are both drugs used to treat fleas in dogs and cats, and they seem to work very well orally to treat other crustaceans in fish. Luferunon inhibits the cycle of the parasite, while nitempiran is an adulticide. In a dog, when you give nitempiran, all the fleas start to jump out the dog in 1 hour or so. It's amazing, and you give it orally. We develop underwater treatment for animals in the water, in large volume tanks, to treat for eye problems with parasites, of course. And right now we are working on oral benzimidazole carbamate to treat from skin and gill trematodes, and also other drugs to treat trematodes and crustaceans. Here, for instance, you have the nitroxylene. It goes into the water, it's water soluble, but it takes a little bit to get completely mixed with the water. So when we have to treat a fish into a big aquarium, we can generate a cloud of the antiparasitic drug, and all the parasites of eyes and gills will die in a few minutes. And how to adapt the present veterinary technology to make it applicable in our aquatic species, as we say. We have many instruments for veterinary medicine, but we cannot use them in our patients because of the size, because of the aquatic environment. How can do? Okay, we adapted, that's something common in Spain, but adapting a regular stethoscope into a esophageal stethoscope is very basic and very useful, especially for a species where you cannot go through, for instance, a turtle through the cell, or a penguin or a sea lion, where it's difficult to reach the heart from the outside. Using a esophageal stethoscope during anesthesia is much useful, and it's very easy to build it. I'll show you some images. Transformation of disposable single pulse oximeter terminals in rectal or esophageal long-life pulse oximeter probes to measure oxygen saturation and pulse in patients. Also show some images too. Use of X-ray films to develop precise anesthesia face masks for aquatic birds, or develop a special stain in the Unipet system to shark leukocyte count. That you all know that sometimes cell counts in fish are not so easy. For instance, here you see the parts to build up an esophageal stethoscope. Very easy. You have the stethoscope, you take the membrane out, and you connect the gastric tube to it, and put a finger of the globe at the end of it. You can put this in the sea lion just through the esophagus in the base of the heart, and you can monitor the sea lion or the turtle or the penguin or whatever animal you want to monitor. And it's much more efficient than the normal stethoscope. Very easy to do. Here you have a pulse oximeter. This is the normal finger detector. Okay, we take it in pieces after. They are discardable. So we, instead of discarding them, we take them in pieces and build something much more useful for our world. The sensor, you should put them one in front of the other. This is the emitter and this is the sensor. What we do is take them apart and put them together. So the light that this emits, this one receives it. But instead of being one in front of the other, they are both together. This thing we cover with resin, and we have a very thin probe that we can use to measure pulse of the animal and oxygen saturation in a small fish, in a small turtle, in birds, or in mammals. That, the digital one of humans, we could never use it. For instance, with all X-rays, and especially those ones that are transparent, or we had not to develop, so it's transparent, so you can see the animal inside. Very easy to build up face masks for birds. I mean, you can, depending on the size of the beak, you make it longer, you make it wider. Very useful as well, and very easy to do. So I just finished with this couple of sentences that imagination is much more important than knowledge, definitely, completely agree, and that logic will get you from A to B, but imagination will take you everywhere you need. Thank you very much.
Any questions for Daniel? Are people awake this morning? No questions yet.
Yeah?
Yeah? Okay, I can't see you, but get a microphone, please. And please say your name and the institution where you come from.
Hi, my name is Chris Brown. I work at the Weymouth Sea Life Park. It was an interesting talk. Last year we had some problems with cow-nosed rays where they came in and they're very infected with leeches, and we tried lots of different treatments to remove them. And one of the problems we had was they're actually on some of the eyes of the cow noses. And I was just wondering if your eye drops, I think it was called...
Nitroxylin.
Yeah, nitroxylin. Would that work to remove the leeches?
I think it could work. I mean, you have to experiment, I guess, but apparently it should cover leeches as well. It depends on the species of leeches that they are more or less resistant, but definitely I will try to see if it works. I cannot tell you for sure, but probably it should work.
Okay, thanks.
Any more questions? Okay, thank you very much, Dania. Very nice presentation.